Hello, everyone, and welcome to today's broadcast. Aurora Kinase drives the evolution of resistance to third-generation EGFR inhibitors in lung cancer. Presented by Dr. Khyati Shah, postdoctoral research scholar at University of California, San Francisco. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Shah. I will now turn the presentation over to her.
Thank you, Howard, and welcome to the webinar on Aurora Kinase A drives the evolution of resistance to the third-generation EGFR inhibitor in lung cancer. The main learning objectives in this webinar is that I will introduce you to the EGFR-TKI, mechanism of resistance as a limiting barrier to the success of the therapy. Also, I will introduce you to the methods or a model system that we use to study the acquired resistance, and a high-content microscopy-based platform that we use to discover the rational combination therapy in case of acquired resistance. Also, I will be discussing about my results, how Aurora Kinase A, we found that it is a synthetic lethal target to overcome the resistance, and the underpinning and the mechanisms of dissecting the residual disease. In addition to that, we also learn about how to design a biomarker in case of the EGFR-TKI resistance. And at the end, I will give the conclusion of my summary. So let's get started with this. With the advent of genomic era, a lot of genetic drivers have been found in case of a non-small cell lung cancer, and majority of them are KRAS mutations and EGFR activating mutations, followed by BRAF mutation, PIK3CA mutation, and the other genomic changes. However, majority of the patients have unknown genetic drivers as well. But for the purpose of this talk, we are just going to focus on EGFR mutant tumors that are shown in the red chunk of the pie. So EGFR activity was identified in 1981. EGFR inhibitors were developed in 1994, and the response to the EGFR was reported in 2004. In that year, gefitinib and erlotinib was approved in 2004. And ever since that, the therapy has seen success, but however, acquired resistance is a major problem to this therapy. As you can see here in the CT scan, at the time of diagnosis, patient present with the EGFR exon 19 deletion, and the tumor has quite you can see the high tumor mass. At the 3 months of the therapy, you can see the tumor size has shrunk, so patient display initial profound response. But upon progression at 20 months, the patient's tumor starts to grow again, and this patient is known as a resistant to erlotinib therapy. And this happens inevitably for most of the tumors. So let's look at what are the multiple mechanisms of acquired resistance. At the cellular level, if we take EGFR activating mutation induces the downstream signaling in PI3 kinase and MET kinase pathway, which leads to cell growth and survival. Hence, EGFR-TKI are designed to downregulate this PI3 kinase and MEP kinase activity. However, the secondary mutations in EGFR occur, which turns on the downstream signaling, such as PI3 kinase and MEP kinase signaling, resulting in cell growth and survival. And these genetic alterations that happens in EGFR are mainly T790M mutation or C797S mutation. Additionally, the bypass signaling such as MET amplification, HER2 amplification, or BRAF mutations are also observed in case of an EGFR-TKI resistant patient. And these also converge to PI3 and MEP kinase signaling, governing the cell growth and survival. Apart from that, the cell state changes or a non-genetic resistance mechanism such as conversion to small cell like or squamous differentiation cell-like phenotype or epithelial mesenchymal transition has been observed. There are also reports which says the NF-kappa B activity or KDM5 or Wnt signaling activity is higher in these patients. And there are chromatin changes that occur when they have no known genetic drivers of resistance. That being said, I'm just summarizing that in the chart over here. As you can see, EGFR T790M mutation is the major resistance mechanism to first generation EGFR inhibitor, that is erlotinib or gefitinib. However, 30% of the patient have no known genetic driver. So hence, first generation EGFR inhibitor are non-selective for T790M mutation, and hence the efforts were done to design the second generation EGFR inhibitor, which had some efficacy towards this resistant mutation, but the toxicity with the afatinib is profound because it also targets the wild type EGFR receptor, resulting in skin rash to the most of the patient. And hence the third generation EGFR inhibitors were designed, which has specificity towards the T790M mutation and the EGFR mutations, but not to the wild type EGFR. And as you can see over here in the waterfall curve, most of the patients respond to the third generation EGFR inhibitor, which have T790M mutation. However, the response are never complete, and most of the patients do progress on third generation EGFR inhibitor as such. Hence, the efforts are done by the Ash-Arishaday group at Stanford to identify what are the genomic driver of the resistance to the third generation EGFR inhibitor by using the circulating cell-free DNA using the CAP-seq method. And what you can see in this pie chart is that most of the patients harbor MET amplification, HER2 amplification, or EGFR amplification with PI3 kinase mutations or single nucleotide variations. However, 35% of the patients have, again, no unknown genetic driver, meaning that the non-genetic mechanism don't drive this, and ultimately it is possible that the genetic mechanism is grown from evolution of this tumor. So this leads us to ask the question, how does the acquired resistance evolve in case of a EGFR-TKI? Now, this is a CT scan from the patient who had T790M mutations at the diagnosis and were pre-treated with the erlotinib, progressed on that. So these patients were put on the third generation EGFR inhibitor, and you can see some response at 4 months, but again, the patient tumor progresses at the 6 months. And in this case, there is a MET amplification upon progression. So this indicates that the tumor is made up of sensitive cells along with the pre-existing genetic clones. When you treat them with the first generation or a second generation EGFR inhibitor, the progression-free survival is almost 9 to 12 months. These patients have T790M lesions and are given third generation EGFR inhibitor, on which also they progress within 10 months of the therapy. And these tumors are immunorefractory, and there is no further treatment options available. Hence, this is a very critical question that we try to address in our lab. So it is critical to model these acquired resistance in vitro. And to do that, we take how we see the clinical scenario. We take a sensitive tumor. So in the cell culture, we take a sensitive cell line. We treat them with the EGFR-TKI, in this case, third generation EGFR-TKI, such as ruxolitinib and osimertinib. And we see majority of the cells die. And at the end of day 3, there are some cells that survive the initial treatment. And this is exactly what it correlates into the clinical setting. These patients develop the residual mass that is tolerant to the drug, but they do not induce apoptosis. And ultimately, this residual disease go on to become acquired resistant, and cells or tumors start proliferating again, and the patient progress on the therapy. And hence, it is very necessary to understand this mechanism of resistance. And to do that, we took 4 sensitive cell lines with 2 different EGFR inhibitor and developed 8 acquired models of resistance. So to indicate here, we took PC9 cells, which harbored the exon 19 deletion, 1975 cells, which harbored the L858R and T790M mutation, and then the HCC827 and HCC4006 cells, which harbor exon 19 deletion. And the resistant cell lines that come out of it is denoted by OR if it is raised by prolonged treatment of osimertinib or prolonged treatment of ruxolitinib. And as we can see in the colony formation assay, that these cells are highly sensitive. Parental cells indicated here in green are highly sensitive to the third generation EGFR inhibitor, but you do see residual cells in this case. When you take the acquired resistant cells, they are highly resistant to both the monotherapy alone. And hence, we ask a question, what would be the good way to characterize this acquired resistant cells? And as you can see here in PC9 cells, which is the parental sensitive cells, the short-term treatment of EGFR inhibitor is sufficient to decrease the MAP signaling denoted by decrease in the phospho-ERK and phospho-AKT and induction of cleave part. But the acquired resistant cells have higher phospho-ERK, phospho-AKT, and diminished apoptotic response. We further characterize this acquired resistant models by performing the whole exome sequencing, and we found no recurrent mutation, meaning that the resistance is not driven by a known genetic driver. The resistance was reversible, meaning that when you withdraw the drug and after 14 days and re-challenge them with the drug, they were sensitive as the parental cells. There is no evidence of RTK activation based on RTK array, and GACA analysis showed that there is enrichment of epithelial to mesenchymal transitioning cells. These cells also had higher NF-kappa B activity as reported by the Bivona group here at UCSF. So we went on to we wanted to identify how to induce the cell killing in these cells, and to do that we developed a platform which is a high content microscopy based platform to do a combination drug screen. So in this screen, we take the sensitive cell line, which is 1975, and a resistant cell line, which is 1975 ruxolitinib resistant cells, and we put seed them on in a high throughput manner in 384 well plate and perform 94 drugs along with a combination with EGFR-TKI. Our readout is DAPI staining, which gives the count of cell proliferation as indicated here in the blue, and YoPro staining, which is indicated in red. YoPro indicates the cell death, and we perform high content automated microscopy and image analysis. And we generate these metrics, which denotes efficacy, meaning that if the drug has, as a single agent, have higher efficacious, then you see effective combo, or they are not effective, that is as an ineffective combo. However, we are interested in a synergy matrix, meaning that both the drug by itself is not effective, but as indicated in the green line, but the combination is able to induce potent cell death. So we take this result, convert them into the synergy score and order them in the ranked sum order. And I'm going to show the results from the screen. So this was performed in 1975, ruxolitinib resistant cell line. And you can see the top two agents that synergizes with the CO1686 and induces the most reduction in cell proliferation in combination with the Aurora kinase inhibitor, AZD1152 and VX680. We further validated this in the long-term colony formation assay. And as you can see here in the panel DMSO, the cells are proliferating. They are AZD19291, which is osimertinib, is also ineffective. And MLN as a single agent, which is also another Aurora kinase inhibitor, is ineffective. But the combination of the both can reduce the cell proliferation. Also, AZD1152, which was the top hit in the screen, did the same thing. The combination is much potent, and so is VX680. So we confirmed our results in vitro and also in xenograft study when we take the PC9 ruxolitinib resistant cells and treat them with the single agent versus the combination, you see potent reduction of tumor volume in case of a combination. So by this time, we know that our combination works highly, but why there is a dependency on Aurora kinase? So to understand that, let me give you a basic of Aurora kinase biology. Aurora kinase is a family of three Aurora kinase A, B, and C. Aurora kinase A localizes to the spindle pole as indicated here by the red color. Aurora A is also involved in regulating the survival during the mitotic delays or error. And it is regulated by a number of upstream factors such as TPX2. So TPX2 binds to Aurora and causes its autophosphorylation. TPX2 interaction with Aurora kinase A also limits the binding of phosphatases, which is responsible for dephosphorylating Aurora A. So meaning that TPX2 is the co-activator of Aurora kinase A. And why is this important? Because in our acquired resistant cell model, we found that as compared to the parenteral cells indicated here in green, phospho-Aurora was higher, or Aurora kinase activation was higher in the acquired resistant cell line indicated here in red. And this happens in all four models. As you can see, TPX2, which is a co-activator of Aurora, is also high in the acquired resistant models versus the parenteral model, meaning that the TPX2 is governing the Aurora kinase A activation. The next question is, what happens if we overexpress Aurora kinase A or TPX2? Does it confer resistance to the therapy? And the answer is yes. So as you can see here in the blue line, is the leg Z control. And when you so 20% of the cells survive upon AZD9291 treatment. When you have Aurora kinase A or TPX2 or a known KRAS G12V mutations, these cells are able to survive better. And this indicates that the TPX2 or Aurora kinase activity is sufficient to induce acquired resistance. So this was very intriguing observation for us. Now, it's sufficient to induce cell proliferation, but is it sufficient to induce apoptosis? That's a question we asked here. And the answer is yes. So the graph here is shown in case of a PC9 cells, MLN itself it doesn't induce cell death indicated by low YOPRO positive cells. But the single agent osimertinib and the combination is able to induce much more potent apoptosis in PC9 as well as in the resistant cells, only in case of a combination. So this leads us to believe that we wanted to identify what are the molecular mechanisms behind the behind this resistance. And to do that, we took the sensitive cell lines and asked the question, there is no target reactivation in case of a combination or the MLN treatment. And the known phospho-Aurora is also not high in the parenteral cells. phospho-BIM, which is also another mediator of apoptosis, and it's been known by the study of Ryan Corcoran and David Costa at Dana-Farber. They indicated that EGFR drives the cell survival by degrading BIM, by increasing the phosphorylation of BIM. So now when you give EGFR-TKI, you should see BIM stabilization and downregulation of phosphorylation of BIM. And that is what exactly we see in the parental cell line in lane 2 and 3. The combination is able to reduce BIM phosphorylation and induces the BIM stabilization. Subsequently, it also causes a stabilization of BAX and upregulation of PAR, meaning that the cells are undergoing apoptosis. However, in case of acquired resistant cells, again, we don't see the activation of phospho-EGFR, meaning that these cells doesn't have EGFR activating mutations. These cells have higher phospho-Aurora, and the combination is able to remove the higher phospho-Aurora in these cells. Also, the Aurora kinase is responsible for the avoiding apoptosis when the cell is undergoing mitosis. And what you can see over from here, that Aurora kinase is known to phosphorylate BIM and regulate the stability of BIM. And in case of acquired resistance, now you can see that the regulation of phospho-BIM is maintained by two different axes from EGFR as well as from the MLN. And both the therapy by itself is not sufficient, but when they are used in the combination, it can downregulate the phosphorylation of BIM, induce BIM, induce BAX, and induce cleave part in case of a combination, in case of acquired resistance. So this is the underlying mechanism of synergy that it induces the apoptotic, it regulates the apoptotic molecule process. So finally, to summarize this section, we can see that the treatment-naive cells, when treated with EGFR-TKI alone, causes BIM stabilization and hence induces apoptosis. However, in the acquired resistant cells, there is a parallel dependency created by phosphorylation of Aurora, which phosphorylates BIM and degrades BIM. But when you use the combination of EGFR and Aurora kinase, you see no phosphorylation of BIM, and hence the BIM is able to stabilize and induce apoptosis. So this is a potent synthetic lethal interaction where the gene A, which is EGFR, is governing the apoptosis, and gene B, which is Aurora kinase, is governing the apoptosis. And when you just inhibit the pathway A, the apoptosis is not abrogated. The combination of the two is required in order to have a synthetic lethal interaction and inducing potent apoptosis. And that is the reason of the potent synergy that we see in the combination as compared to combination therapy alone in these acquired resistant cells. So moving on to asking a big question, how does the acquired resistance evolve? So just to give you a perspective of what's been known in the literature until now, there are pre-existing T790M mutations that happen and which governs the acquired resistance to the first generation EGFR-TKI. Or there is a persister evolution phenotype, meaning that these cells are initially sensitive. They enter into the residual-like state, which have the T790M clone, and develop the T790M positive clone. But this is in case of a first-generation EGFR inhibitor. It is also known that prolonged treatment of EGFR inhibitor causes this residual cells that are called DTP, or a drug-tolerant persister cells. And when these cells are kept in drug, they start cycling and they become DTEPs, which is the acquired resistant cell line. And Jeff Settleman group in Genentech had shown that these cells are DTPs have higher chromatin condensation in them. But until now, there is no clear answer how the cells transition between the sensitive state to drug-tolerant persister or residual cells to the acquired resistant cells. Also, there is one study from the Altruger group here at UCSF where they expanded the sensitive PC9 cells from a single cell clone, and they made 17 different cell lines and kept them in the culture long term for about 52 weeks. And they asked the questions, the cell lines that come from these, what are the genetic changes in them by performing the whole genome sequencing. And as you can see here that the cell lines are on the y-axis and the changes that happens in the genomic changes are on the x-axis. And you can see most of the cells have either EGFR mutations in them, MET amplification, BRAF, RAF mutations, and RAS activation, or MAP kinase 2 mutations. And there is also high dependency on AKT or PIK3CA mutation, consistently what has been reported in the literature. However, this is a very long-term treatment, and we don't know what is in between. And hence, so it leads us to ask the question, can residual cells give rise to acquired resistance? Also, do these residual cells have targetable vulnerability that they can be targeted? And does the suppression of the residual cells prevent the emergence of acquired resistance? And hence, we went back to our model of acquired resistance and the residual cells. So in this case, from day 3 to day 9, we have our residual cells. And we asked the question, what are the changes that happens upon the drug treatment over a period of time? And to do that, we performed the Western blot analysis at the time that are indicated over here in the parental 1975, which is a sensitive cell line, and in the 1975 AR, which is the acquired resistant cell line. And clearly what you can see over here is that the treatment of EZD9291, which is the osimertinib, is able to reduce the phosphorylation of EGFR throughout the treatment duration. The target activation is not taking place. However, the TPX2, which is the co-activator of Aurora Kinase, starts to go high at the end of 24 hours, and it's maintained throughout the residual phase and in the acquired resistant cells. Also, it increases, TPX2 is known to stabilize and activate Aurora Kinase, and hence we see the temporal activation of Aurora Kinase, followed by TPX2 activation at 24 hours, and these cells in the residual state have higher Aurora kinase as well. In terms of phosphorylation of BIM, these cells are able to suppress the phosphorylation of BIM, and when the Aurora kinase activation occurs, these cells now is able to phosphorylate BIM and destabilize BIM. So in the sensitive cells, you can see that in the sensitive phase from day 2 to day, at 4 hours to day 2, these cells are able to induce apoptosis, but from day 2 onwards, they enter into the tolerant cells, and they have abrogated the apoptosis by destabilizing BIM. And hence, we can use this model of residual cells to study the residual disease in clinic. And so we asked the question, does activation of phospho-aurora happens with the other EGFR-TKI as well? And the answer is yes. As you can see here in the graph, that PC9 that are sensitive cells, when they're treated with the indicated first generation, second generation, and third generation EGFR inhibitor over here, respectively, is able to induce it's able to induce the Aurora kinase activation. TPX2 is also high in them, and they are able to abrogate the apoptosis by destabilizing BIM. And then the question is that how can we prevent the emergence of acquired resistance? So this is performed by prolonged treatment, day 90 treatment of the individual therapy. And what you can see over here is that PC9 cells without any treatment proliferate, but and so as in presence of Aurora kinase inhibitor alone. In case of a single agent EGFR therapy indicated here in orange and in green, the cells enter, there is an initial response, and then the cells enter into the residual phase, and then after day 30, they start growing back again. But when you treat them upfront with the combination therapy, it's able to reduce the cell proliferation by in a profound manner, so that you don't see the activation of cell proliferation and cell growth. The other question is, what happens when we have the residual cells? So here we made the residual cells, and then we asked the question, what happens in presence of combination therapy? And what you can see in the red and the pink line is that the combination is able to remove these residual cells, meaning that it has a potential of eliminating the residual disease as well. To confirm this, we took the PDX samples that were derived from the EGFR L858R mutation tumor. This patient was initially responding to erlotinib, but however, they developed the residual disease, and that's where the biopsy was taken and the patient-derived tumor was made from these. This residual tumor doesn't have the known genetic aberration, such as T790M mutation or any other. And in the paper, in the cell report paper, Colin Blackley shows that these have higher NF-kappa B activity as well. So we use this PDX model to ask the question, is it sensitive to the third generation EGFR inhibitor alone? And what is its efficacy in the combination? So as you can see over here is that the PDX itself grows when there is no treatment given, indicated in the blue bar. When you give Aurora kinase inhibitor alone, in the green bar, the tumor is still proliferating. Single agent osimertinib, which is the EGFR-TKI, is also not able to reduce the cell proliferation, although these patients have EGFR mutations in them. But the combination does much better job in preventing the tumor growth of the PDX, meaning that the combination is highly synergistic. And again, we performed the molecular signaling studies by performing immunoblot. And we show here that EGFR inhibitors alone induces TPX2, and the combination is able to abrogate the TPX2, which is a resistant marker, and phospho-Aurora. These cells also have diminished phospho-BIM, and there is a BIM stabilization that happens in the combination, along with the cleave-PARP induction. So this combination is cytotoxic as compared to cytostatic. Also, we identified TPX2 as a marker of resistance, meaning that the patient who progress on EGFR-TKI or the tumors, PDX tumors that progressed on EGFR-TKI have higher TPX2 staining, as indicated by the IHC staining over here in the picture. Also, TPX2 is highly upregulated in the patient who progressed on erlotinib. So shown here in the panel are two patients which were biopsied at the time of diagnosis, indicated by A, and at the time of progression, indicated by B. So as you can see, in 6 out of 9 patients, the TPX2 went higher in case of a resistance, meaning that TPX2 is a marker of therapeutic resistance. And as you combine, there is a statistically significant difference in the TPX2 IHC score in the patient who progress on first generation EGFR inhibitor. Also, we took the 3 samples that were identified from third generation EGFR inhibitor. And as you can see here in the IHC, the pre-treatment and TKI resistant tumors, TKI resistant tumors have significant higher IHC of TPX2. So this makes us believe that TPX2 could be a potent biomarker for the therapy resistance. So to summarize my findings so far, the treatment induces the activation of Aurora kinase A, which is associated with the resistance to EGFR-TKI. TPX2 plays an important role in Aurora kinase A activation, and it contributes to the acquired resistance to EGFR-TKI. TPX2 can serve as a biomarker of treatment of resistance. And Aurora Kinase A activation deregulate the BIM-mediated apoptotic pathway in the residual cells, and hence the combination is, and hence the inactivation of Aurora Kinase A serve as a synthetic lethal strategy for enhancing the responses to EGFR-TKI. With that, I would like to thank my lab and my PI, Bandyopadhyay, and the lab members, along with our awesome collaborators from Bivona Lab who assisted in my studies, UCSF Pathology Group, Gregor Kings for performing the pathology on the samples, Goga Lab, Dumont Lab, McCormick Lab, and my funding, and UCSF Helen Diller family. So thank you all. And this is accepted in the Nature Medicine. So this paper should come out in a couple of months. And please cite our work and let me know if you have any questions. Thank you.
Thank you, Dr. Shah, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Now let's get started. Our first question is, can genetic and non-genetic mechanisms coexist? What is the clinical implication?
This is a good question. And yes, the answer is that genetic and non-genetic mechanisms can coexist. We think that Aurora kinase helps in the evolution of the resistance mechanisms, and ultimately the clone that is fit to survive will survive in the tumor and give rise to a genetic resistance. It's been shown that multiple genetic drivers as well as non-genetic drivers are present in the clinical patients who progress on EGFR-TKI, and hence it is important to study the genetic and non-genetic mechanisms.
An innate resistance of EGFR-TKI is always the genetic mechanism, or do non-genetic mechanisms exist in such a case?
So for the innate resistance mechanisms, other than T790M pre-existing clone, there is no other known genetic mechanism that can give rise to innate resistance, and hence it is believed that the non-genetic mechanisms exist in case of innate resistance to EGFR-TKI.
I would like to once again thank Dr. Shah for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2019. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.